One of the most important biochemical applications of the Claisen condensation occurs in the biosynthesis of fatty acids. Fatty acids are very long-chain carboxylic acids, which consist of a polar end, where the carboxylic acid functional group is located, and a nonpolar end, characterized by a very long chain of generally saturated, singly bound, or sometimes doubly bound carbons. The synthesis of fatty acids requires the formation of a lot of carbon-carbon bonds, and it's a Claisen condensation that establishes these carbon-carbon bonds. Each 2-carbon unit that we see within a fatty acid chain is derived from the biomolecule acetyl coenzyme A, sometimes abbreviated as acetyl or acetyl-CoA. And each of the bonds between these 2-carbon units is formed through a Claisen condensation. In every Claisen condensation, we have a nucleophilic ester and an electrophilic ester, and we're going to look at both of these on this slide. One of the first things to note is that nature makes use of not esters, not alkoxy esters, but thioesters, and more specifically, the molecule malonyl coenzyme A, which you see on the right-hand side of this reaction scheme, is nature's Claisen nucleophile. Malonyl coenzyme A itself is actually generated through an addition reaction that looks a lot like an aldol-type addition involving an enolate and something that looks like carbon dioxide. So in this reaction, we establish a bond between the alpha carbon of acetyl coenzyme A, here I'm highlighting that in red, and the central carbonyl-type carbon in bicarbonate. And what it looks like we've done is the addition of an enolate to carbon dioxide to form a product in which we have a thioester and a carboxylate group at the beta position. This is malonyl coenzyme A. Acetyl coenzyme A, which is this molecule here that's ultimately the starting material for the synthesis of malonyl coenzyme A, is nature's Claisen electrophile. Even though acetyl-CoA is enolizable, nature is able to orchestrate this in the active site of an enzyme so that it doesn't get deprotonation and it undergoes nucleophilic acyl substitution instead. Both bind to a protein called an acyl carrier protein before reacting, and we're going to abbreviate that in the figure below as ACP. This is just a protein fragment that helps with molecular recognition of the nucleophile and electrophiles so that they're steered into the right positions before they react within the enzyme's active site. When the malonyl ACP on the left reacts with the acetyl ACP on the right, carbon dioxide is lost from the malonyl ACP, and this nucleophilic carbon forms a bond to the electrophilic carbonyl carbon of acetyl-CoA. So ultimately we get nucleophilic acyl substitution at the carbon highlighted in blue and an acylation at the carbon highlighted in red. And notice the analogy to the laboratory Claisen condensation. This effectively is a Claisen condensation where the product we get is now not a beta-keto ester, but a beta-keto thioester. The reactivity is exactly analogous to the Claisen condensation. And one quick note about the decarboxylation. Notice that if we eliminate CO2 from this intermediate by kicking off this carbon as a leaving group, the molecule that we end up with is the enolate of acetyl-CoA. So really, the use of malonyl-CoA as the nucleophile comes down to this ability to decarboxylate, eliminate CO2, and generate an enolate in the process. This isn't exactly how it works mechanistically, but this gives you a good idea of why malonyl-CoA is used as the nucleophile in this context and how the electrophilic neutral acetyl-CoA combines with the enolate of acetyl-CoA to form this product. One other thing we're not seeing here is that the ACP thiol, which we can abbreviate as HS-ACP, is kicked off as a leaving group in a beta elimination type step. Let's take a quick look at the mechanism of this reaction in an enzyme's active site in a little more detail. And I'm going to represent a general acid within the enzyme using this abbreviated structure with HA representing the acidic group. This might be something like an aspartic acid residue, for example. In the first elementary step, decarboxylation is accompanied by protonation. Malonyl ACP undergoes a beta elimination of carbon dioxide and simultaneous protonation to form an enol intermediate. On the last slide, we thought about this as an enolate, but under biological conditions, we want to avoid that reactive enolate intermediate. And so in this first step, a nucleophilic enol of acetyl-CoA is formed. At this point, the electrophilic acetyl-CoA enters the picture, and we can now think of the conjugate base of the acid, A-, acting as a general base to promote the nucleophilic addition of the enol to the carbonyl carbon of acetyl-CoA. We've now generated an oxyanion, which is poised for beta elimination, and the general acid catalyst that we now have can assist in this by protonating the leaving group as it departs. This third step generates the neutral beta-keto thioester product, as well as the ACP thiol. From the broader perspective of fatty acid synthesis, what this reaction has done is elongated the thioester by establishing a key carbon-carbon bond between the alpha carbon of acetyl-CoA and the carbonyl carbon of another molecule of acetyl-CoA, in essence lengthening the fatty acid chain by 2 carbon units.